Okay, great. Hi, everyone. My name is Trevor Pugh. I'm a senior scientist at Princess Margaret, and I direct the genomics program here at OICR, as well as the Princess Margaret Genomics Center, which houses the Brain Single Cell Initiative, which is one of the sort of genomics cores that offers a lot of the single cell technologies we're going to talk about today. So that's the logo in the middle. So if you want to do some single cell work, you can check out the PMGC website. Yeah, so I'm going to give my role in this workshop is really to give you the technological overview, some of the conceptual shifts from bulk to single cell sequencing, and introduce some of the sort of single cell technologies that are current today, but the field is moving extremely quickly. So what is current today may not be what's current sort of 6 months from now when we give this talk again.
Sorry, we're getting some echo. Is anyone in here connected to the Zoom?
Oh, sorry.
Okay. Yeah, so the learning objectives for this opening talk are really about understanding the conceptual shift from bulk RNA sequencing, which many of you may have already interacted with, and what are some of the assumptions and technologies that are different moving from bulk to single cell. Learn about the types, parameters, and some of the trade-offs of the single cell sequencing technologies. And definitely in my role running a lab that does single cell sequencing, the number one question we get asked is, which technology should we use? Do we need spatial? Is it droplet? There's 5' N, there's 3' N. Which technology should we use? And it's really highly specific to the scientific question that you're trying to answer. So we'll go through some of those considerations that we'll often go through speaking with collaborators, trying to understand their scientific goals and line up those scientific goals with an appropriate single cell technology. My lab's cancer lab. I'm at Princess Margaret. So I'm going to use cancer sort of as an example of going through that decision-making process. And then, of course, this whole workshop is all about spatial. So we'll talk about some of the Visium and the move from Visium regular to Visium HD and some of the caveats with that technology. So the first point is we're all made of cells. So cells are the functional unit of life in general. But they're not just a grab bag of M&Ms. They have structure, they have form. Cells depend on one another. If you're doing droplet-based sequencing, your data is going to look more like a bowl of M&Ms. You're reaching in and grabbing a bunch of cells. There's a sampling experiment there. Sometimes you get more red M&Ms, sometimes you get more green. Tissues are exactly the same way. If you're digesting them and pulling out cells, there's a bit of a sampling effect. The spatial technology is a little bit different because they keep that spatial knowledge intact. And the opportunity, especially with the spatial technologies, is the opportunity to try to understand some of those cell-cell interactions or even just cell-cell dependencies. Single cell is also not new. It's kind of new in genomics, and it's really undergone a very recent technological boom. But I've actually just shown on the left, top left, that's actually my karyotype. So that's one of my blood cells I dropped on a glass slide. In the old-fashioned days, it would land as like a pile of chromosomes, and you'd literally use scissors and glue to do what's called a paste up. You'd cut out your chromosomes, line them up, and order them in that nice way. So they don't come like that in a cell, but that was like my first experience with single-cell genomics. Of course, it's all done by computers now. And then the bottom left is a single in situ hybridization for a single viral RNA transcript. So we're trying to say, is that virus expressed in a single cell? So it turns dark purple. That concept has really aged very well, except now we can do every single chromosome in every single cell. So that's the top right. And we can do most transcripts in pretty much every single cell. And soon, I actually believe it will be every transcript in every cell. But conceptually, it's really the same. It's what's going on in a single cell. What are the somatic alterations? So in the upper right, that's a cancer sample. So there's gains and losses. So we're trying to answer more or less similar questions, but at greater resolution and a greater breadth, going genome and transcriptome wide. The other opportunity is getting away from averages. So bulk RNA sequencing, you take your sample shown on the left, we grind it up, we sequence the RNA, but as a result, you're getting an average of all cells in the sample. A good example is the Cancer Genome Atlas. It was a huge initiative to sequence thousands of tumors from as many different cancer types as possible. It set out to understand cancer, but now all that data actually gets used and reused to look at immune profiles, stromal interactions, stromal infiltration, because all those cells kind of came along for the ride. So even though the initial goal was to grind up and sequence tumors, it's actually a great resource to look at some of the other non-cancerous cells that are available in that sample. And since TCGA, which is a bulk genomics project, we've now moved to really looking at the same, very similar questions, but at the single cell level. So that's really what the lower right is showing here. Really looking at individual gene expression in individual cells. And looking not just at cell identity, but also the cell state. And we'll circle back to this when we get back to the cancer example in brain tumors at the end. I put this title in quotes. It's literally a copy and paste out of the abstract for this really great paper that outlines at that point in time, which was a few years ago now, 2019, all the different technologies and approaches to understanding single cell data. I added some red labels at the top, lineage, state, and trajectory. So I sort of broadly thought of these as sort of big conceptual areas that single cell data can be used to address. So understanding how cells and tissues develop. How do you go from a stem cell into a fully formed organ? Cell state, if you know what a cell is, T cell, B cell, what is it doing? Is it cytotoxic? Is it a memory T cell? Is it a regulatory cell? So beyond just what is the cell label, but actually what is it doing? And then trajectory within a tissue or within a sample. Is there a sort of a developmental lineage, especially important in cancer, where you sort of start with an early cancer that actually spins off all these progenitors? So using single cell data to sort of understand both abnormal and normal cell development. And I've just put in the red, we're going to focus on two boxes in this very complicated figure. Spatial position, that's sort of the big benefit of the Visium platform, and single cell RNA sequencing. I put this title also in quotes, also a copy and paste right out of their abstract. Even though this slide is a few years old now, this trend has really held. So this paper really outlines, really from the early days, actually some of it done by Norman Iskov here in Toronto, doing single cell sequencing of RNA in a tube, really one cell at a time, sort of expanding that into plates and microfluidic platforms, the high throughput robotics letting you do hundreds of cells at a time, the advent of droplet-based transcriptomics, that's moving you up to thousands of cells at a time. And where we are now looking at complete tissues, is now we're looking at hundreds of thousands of cells simultaneously, all with kind of the same goal. How do you measure and understand what a cell is doing and some of the interactions of that cell next to it? And this slide sort of shows that number of cells increasing over time, but also calls out some of the technologies that were key to that year-over-year exponential increase. The new revolution we're going to talk about today brings spatial technologies. The marketing material looks like the left. You get these beautiful swirls of really nice cell clusters and beautiful sort of blood vessels and stromal infiltrates and immune cells. But computationally, it's much simpler than that. It's just a table. It's a table of every cell by every cell and a distance between each of them. So certainly the marketing image is helpful, but really you're still doing single cell sequencing. You just have an additional piece of metadata that just says this cell is however many microns away from this other cell. So I encourage you, especially as bioinformatics analysts, think more about the cell distance matrix rather than the structural position as it appears on the slide. I'm going to show a lot of these like nicely colored, spatially organized cells, but computationally, it really is just an XY axis. If you're really lucky, you might have 2 or 3 sections in a row. You'll have like a Z stack of 3 different distance matrices. Also very interesting computationally. But really, the only added new piece of data is this distance matrix, which you can do a lot with. The other exciting part is really being able to look at complete organs, at least in the case of a mouse. So this is from 10x Genomics Mouse Tissue Atlasing Project. That's a mouse pup. But it was really the first time I'd ever seen on a single slide an entire organism fully profiled for every cell using a gene panel. So it's not like they did every cell and every gene. That technology at that point was a 380-ish gene panel. But this is the type of research that's now possible. You can really look at complete developmental stages across complete organisms that can fit on the glass slide. So obviously not going to do this in human, even with a big human organ, but you can be really quite creative in terms of how you organize even samples or small model organisms on a glass slide, and then leverage that distance matrix to understand interactions within an organ, but also between organs potentially as well. And some of the questions that you can answer, I outlined from this review on the right-hand side. Some of the questions that are used using this spatially organized data. So disease research, tissue development, I think is probably the most interesting and probably the most powerful use of this technology, because you can look at the same sample or the same genetic model over time. So those serial samples are very powerful. And then tissue architecture, how do cell dependencies or cell distances actually change during development, during treatment? Serial biopsy is also very interesting. So we're really trying to think of how to leverage not just the RNA sequencing data, but that distance matrix as well. So how do you make these images? There's a lot of ways to do it. We're going to mostly talk about 10x's offering, but there are a lot of other companies that are coming online to offer very similar technologies. This slide really shows sort of the three broadly, the three broad strategies that companies have commercialized. The first one is essentially an array, so it's a capture-based. At the top, the capture-based method, you basically put down a carpet of oligonucleotides and a patterned array. So you actually don't necessarily know whether your cell is going to land perfectly on that spot, a little bit to the right, a little bit to the left. Sometimes a cell will sit between two spots. So it's really saying these RNAs are at this position on the microarray, but it doesn't necessarily map it perfectly to a cell. And that's actually a feature of the Visium platform. It's actually just a gridded array. You actually have no control over whether your cell is going to land perfectly in a spot. The older Visium technology, multiple cells often land on top of a spot. So it has a downside in that it's not true single cell. It's more like single cell resolution is how it's often described. But it does have the power of true RNA sequencing, capturing the RNA on that gridded slide, and then start to count transcripts. The other two approaches, in situ sequencing and in situ hybridization, these typically are true single cell. So in situ sequencing is essentially taking the RNA as it exists within the cell and sequencing it. The key to this technology working is the cell segmentation. So it's really trying to understand, what does the cell outline look like? Often this means taking an H&E or having a different stain to outline the cell membrane. If you're able to do that, then you can actually localize cells truly within a cell. And then in situ hybridization, this is the 10x Xenium platform. Same kind of idea, in this case using a gene panel to hybridize two transcripts of interest. This last approach is the most sensitive, but it's also the most restricted in terms of the number of genes. So if you have a gene panel or you really have a narrow scientific question, very interested in a certain pathway or certain cell type, really the bottom probe-based approach is very powerful because you can really have that sensitivity to pick up every transcript of interest. Whereas sort of row B, that's more of a broader search for all transcripts across all single cells, but has the downside of limited sensitivity. So here's how 10x Genomics is commercialized, those concepts. So the top one is the droplet-based method. You take your tissue, you don't care about the spatial orientation, you digest it, capture your cells in single droplets, and then do your molecular biology reaction within a droplet of oil. The middle one is the Visium platform. So this is this gridded array of oligonucleotides. They've just sort of upgraded Visium from low resolution, which is sort of 8 to 10 cells per spot, to Visium HD. And the reason it's HD is the spots for the first time are now the same size or smaller than a cell. The marketing material says you can do subcellular resolution. Challenging to do, unless you have a really big cell, you might be able to do that. But it really is approaching true single cell resolution, but has the problem of the cell segmentation issue. The carpet of oligonucleotides are evenly spaced. It's like your gridded array. And same sort of problem. Is your cell going to land perfectly on a spot, or is it going to land awkwardly between spots? This, of course, happens. Then the last one, Xenium, this actually, I'll show some of this data in just a second. This actually uses a cell map. So you do a stain just to get the cell outline first. And then you do cell segmentation to really say, here's the outline of every single cell. And then you map the RNA transcripts into those cells. So the question I get the most, how do you choose? There's lots of tools, and my recommendation is don't get too enamored with the technology. Don't put the method first. Formulate your scientific question first, and then see which technology best serves that exact question. And sometimes you'll have 3 or 4 questions, and the question then becomes, can you afford 3 or 4 different technologies, or is there one sort of middle technology that best serves multiple questions at once? So these are sort of the 6 areas I kind of think through when discussing how to design a single cell experiment. Do you need all the genes? So gene throughput? Do you need something like Visium or droplet-based method where you try to capture as many native RNA transcripts as you can? Or do you really know the gene set you're going after, and can you do a targeted panel and benefit from that increased sensitivity by looking at fewer genes? Sequence information, do you actually care what the sequence of the transcript is, or you just want to count it? I'm in a cancer lab. I care a lot about the mutation, so I really want to know what sequence is there. And that, in the early days of single cell, really pushed us towards true sequencing-based methods and not looking at targeted panels. We used, in the early days of single cell, we used droplet-based methods, but that program, especially in brain cancer, has now evolved into a spatial sort of a spatial transcriptomics project. So our brain cancer program kind of evolved over time when we were focused on mutations, and now we're more focused on gene counting. So we've actually changed technologies over time. Sensitivity, do you need every single transcript? Are there markers or canonical markers really well established in your field? Immunology, a great example. There's all these CD markers, super important. I think a lot of our collaborators are kind of disappointed at how hard it is to find a lot of those transcripts. A lot of those markers actually are not expressed at very high level at the RNA level. They're very strong at the protein level, but they don't always show up in single cell. So it's really a question there. Do you need that sensitivity to pick up those rare canonical markers? Or can you use gene pathways or gene sets that are associated with your marker to infer cell identity? Resolution, do you actually care about single cell and the single cell resolution and the spatial organization of cells? Or can you tolerate having the potential that a cell is landing between 2 spots or just being digested using a droplet-based method? So sort of think through, does your scientific question truly require single cell, or are you able to aggregate multiple cells together? If you have a cell line model, if it's broadly homogeneous, probably don't need single cell resolution. Probably the droplet-based method might be a better choice. Area size, do you really need really, really small spots that are smaller than a cell, or can you tolerate aggregation or averages of multiple cells? And probably the defining feature is feasibility. What is the nature of your tissues? Are they fresh out of the operating room, not frozen? They can go straight to your genomics core and get profiled? Or are these like 20-year-old formalin-fixed paraffin-embedded tissues where the RNA is degraded? We've worked with all these things. You need really different technologies. And probably my number one recommendation is spend a ton of time up front optimizing that step from the cell into the single cell platform. So how do you collect the sample? How do you make sure your cells are viable? Can they be frozen or not? Kind of depends on which technology you're using. Are they going through a flow cytometer? If they are they coming out viable or even intact on the other end? So we've had some collaborations. They look great on flow because you're just counting the cells. The cells were dying as part of the flow, as part of the sort. So spend like half your time really getting that upfront connector from patient mouse plate into the single cell experiment. That's really the number one predictor of whether you're going to have good quality data. Yeah, I guess maybe I'll just take a quick pause there. Any quick questions about sort of that overview, the array of technologies that are out there before I dive into a cancer example? I don't see any online. Okay, I'll move on. So
We do. Just to let you know, the microphone system in here isn't working, so you can put that down. Just speak up a little. That would be great. Thank you. We're gonna fix it over the break.
Okay, good. Am I loud enough? Just like this?
Yeah.
Okay.
Diaphragm.
Okay, so this is a picture of a tumor, or actually an artist painting of a tumor. It's really showing, the point of this is to show the huge variety of different cell types that exist within a tumor beyond just the cancer cells. And I usually try to differentiate tumors, which I think of as masses of cells, from cancer cells, which are the true malignant abnormal cells that inhabit these sort of rich and highly varied tumors. The other challenge with tumor sequencing is they tide over time. So sometimes the cancer cells will expand. If there's an immune response, sometimes immune cells will expand. And we are interested in specifically the population of cells that are the cancer stem cells. So we're trying to understand the very, very earliest, or that top of that lineage tree, the cells that give rise to the bulk tumor and encourage or exclude the other normal cells to populate the tumor. So here's an example. This is actually a project we did with Gary Bader and Peter Dirks, trying to understand these cancer stem cells. And the idea was cancer stem cells are resistant to chemotherapy. So you treat the bulk tumor, you kill off the cancer cell, most of the cancer cells, but these CSCs, cancer stem cells, remain, and then they regrow and repopulate. And there's very strong genetic evidence that this happens in glioblastoma because the relapse tumors have totally different mutational spectrum from the primary tumors. So something is really happening from that initial debulking to the regrowth. And the whole idea behind the study was, let's understand the cancer stem cells, come up with therapies to take them out. So even if the bulk is intact, there's no way for it to repopulate itself after treatment. And if you want to read the paper, it's published 3 or 4 years ago now. So to start, we use droplet-based methods. We use the 10x Genomics 3' platform. That was what we had available at the time. We also tapped into a cell culture system that both Peter Dirks and Sam Weiss in Alberta had established. So they both take these tumors at the top, digest them. One of them flow sorts them. One of them puts them in culture that is specific for promoting growth of stem cells. They grow them up. And then we were interested, first off, how are these two culture systems different? But then also, what are the shared RNA or the shared transcriptional pathways that are common across glioblastoma stem cells? So the first question was, or first finding was, they're all totally different. And this is actually held up over and over again. When you take a tumor and you sequence the RNA and try to cluster them all together, the normal non-cancer cells all cluster together. So everyone's T cells are more or less the same. The vascular cells are very similar. But the cancer cells, as shown on this plot, all form their own transcriptional clusters. Even when you had multiple tumors or multiple lines from the same tumor, they didn't always cluster together. They were always near each other and they shared the greatest transcriptional similarity, but they never perfectly overlapped. So that was our first lesson. These cancers have gone so far off the rails, they really are at a very high level transcriptionally distinct. So the question we wanted to answer was really, do they share any sort of common biology, even though at a very high level they're very distinct? So the first finding was, yes, while they weren't exactly the same, they all existed on this injury response developmental gradient. So these are all of the glioblastoma stem cell cultures ordered, and they're all scored using 2 gene sets, an injury response gene set and a developmental gene set. And you can tell that not every single cell from every single GSC was at one spot. It sort of had a bit of a distribution around it. But if you sequence enough of these GSCs, you can actually characterize the entire transcriptional gradient. This was sort of their first look into trying to understand what had then been established as canonical transcriptional subgroups in the primary tumors. And what we were seeing here is actually those groups weren't totally distinct from each other. There was actually sharing across the stem cell compartment. So these stem cells were existing on this gradient and they were spinning out, maybe biased towards one subtype versus another, depending on their position on the gradient. But it was the first time that we really thought, instead of trying to come up with 4 distinct groups, say, no, this is actually a continuous gradient rather than 4 distinct categories. And this is the model we came up with in that paper, where we said all the stem cells exist on this blue gradient. So there's a developmental and injury response gradient. And then they're fountaining out a third population of cells. And this is actually the bulk of the tumor. These are the astrocyte lineages. And we did some trajectory analysis to really show that the stem cells were the starting cell of origin, but then they were fountaining out the bulk tumors that were also distinct from the stem cells. And if you're interested in the tools, we use SC-Velo. We actually tried a bunch of these. This is a very hot topic lately, is trying to bake off all these different velocity tools. This one worked well in our hands. But especially if you have a really nicely controlled experiment of multiple samples, I think that's where these trajectory algorithms really shine. So since then, we wanted to ask the question, okay, we know what these glioblastoma stem cells look like in a dish, but what do they look like actually in primary tumors? So we had some model organisms, we understood the gradient. We now want to understand, are these stem cells just sitting as islands radiating off those astrocytic cells, or are they themselves have a gradient as glioblastoma sort of threads through the brain? It's kind of a feature of that cancer type. So this is work from Shamini, who you're going to organize this whole thing, or most of this. She's going to be sharing some of her data from this type of project. This is really our first Xenium run ever applied to a primary brain tumor. This is a glass slide with two tumors sectioned onto it. So you can see these little tiny pieces of tissue. And this is the hematoxylin eosin same tissue. So you can sort of see this is what pathologists will usually look at to make a diagnosis. You can tell grade. In this case, it was a primary surgical specimen. And in this case, we applied a 300 gene targeted Xenium panel to it. And so this is the exact same piece of tissue, one section down, and now we're just counting transcripts. So 200,000 cells, that was way more than we'd ever looked at before using a droplet-based method. I think we looked at 3,000 to 4,000. So this was like multiple orders of magnitude more cells, but way fewer transcripts. We only looked at 266 transcripts. Before we were looking at 10,000 transcripts per cell. Good news is most of the transcripts landed within cells. This always happens. You get some transcripts kind of floating outside. You can calculate cell density, and you get these very satisfying sort of QC plots. How big is your cell? How many genes do you have per cell? And then now there's the opportunity to start labeling these cells as well. So she ran a clustering algorithm. You can tell there's all sorts of cells inhabiting this tumor, very similar to the artist rendition diagram of this earlier. But the nice thing is when you if you ignore the spatial matrix and just look at the RNA sequencing data, you really start to see some patterns, which I've shown here on the right. And really, there's that big multicolor one. That's the tumor. Those are cancer cells with all the different subclones. And then two of them are totally different, and they don't look like cancer cells at all. And if you look at the cell markers, the top one, very likely to be immune cells, expresses a lot of the genes associated with immune cells. And then the bottom one are likely blood vessels, a lot of the vascular VEGF type transcripts being associated with those genes. So it's really for the first time where you can take these super complex mixtures of cells, computationally deconvolute them, and really ask really focused questions on what's happening in the vasculature, what's happening in immune infiltration, and what's happening in the tumor. And I show that as an example. Lots of groups are doing this and lots of different cancer types. This is a sort of a survey of that a couple of years ago. Basically, every single cancer type has a reference data set of some sort that has some sort of single cell data. So my second recommendation after doing a lot of lab optimization to get your protocol to work is plug into all of this public data. It's really rich and diverse, and it's really an opportunity to merge a lot of your locally generated data with very similar data that was generated for different experimental reasons. So usually the first question I usually have when we get a data set is, does it look like glioblastoma? Does it look like other tissues that have been profiled using the same or similar technologies? What I showed on the previous slide were all cancer data sets. An equivalent and probably order of magnitude more greater research intensity is just focusing on just healthy development, healthy cells. So there's and you'll probably come across in the literature this concept of human cell atlases, grinding up healthy tissues and just trying to understand healthy tissues and how they develop over time. Extremely valuable for cancer researchers because we want to know why cancer cells have gone off the rails. And this is really the opportunity to integrate cancer data sets and healthy data sets to understand healthy cell development, and then at what point in that development has something gone wrong and become a tumor. So same sort of thing. There are probably hundreds of these data sets now. I definitely, even before you generate your own data, do a survey and see what data are already out there that you can download and build right into your analysis plan. And this is a really great example. I think Allen Brain Math's probably done this better than everybody. They've not just focused on transcriptomics. They'll take a sample, section it multiple times, and run multiple orthogonal assays, a proteomic assay, an RNA-seq assay. They do a really great job of spatial mapping and understanding those H&E slides I showed earlier. So if you're working in brain research, Allen Brain Atlas is a really great source of trying to understand this relationship across different layers of biology, not just the RNA itself. Also very powerful as a validation set. So if you do your own local discovery or you have a model organism, it's a chance to go look in mostly healthy brain tissue to see, do you see that same pattern there as well? This is my last slide. So I hope you've sort of gathered there's a plethora of single cell technologies. Don't put the method first, put your scientific question first, and then map the technology to your question. Totally new types of questions can now be asked because we can get away from these bulk technologies that averaged out signal. And even in really complex brain tumors, you can really start to do deep dives on really rare cells of interest, like glioblastoma stem cells. You can measure the same biology different ways. It doesn't have to be RNA. You could measure it using protein. You could do total RNA sequencing. You can do a panel. The biology is the same. So if the biological signal is strong enough, you should be able to see it given two largely equivalent technologies. And that's really where being very specific with your question is so important. Multiple cellular components can be queried from one cell. So you can ask cancer cell questions and immune cell questions and vascular questions all in one experiment. You make one data set, analyze it 4 or 5 different times, and think of that sort of 6 square table, all the different features that kind of go into choosing a technology. A lot of it flows from feasibility. Make sure you have samples that are actually amenable to single cell and spend a lot of time ensuring that connector from patient or model to your genomic sequencing platform is heavily, heavily optimized. So that's my last slide, and happy to take questions if there's time.